Before going to irreversible cell injury, we want to recapitulate the changes we have said during reversible cell injury. As there were reduced ATP formation by oxidative phosphorylation, reduced function of sodium potassium pump that causes entry of sodium and water into the cell, causing cellular swelling. Within the cell membrane, we have said there were bleeding, blunting and distortion of the microvilli. Within the ribosome, there were reduced protein synthesis as a result of swelling and detachment of ribosome from the endoplasmic reticulum. Within the nucleus, there were clumping of chromatin as a result of acidic environment caused by lactic acidosis through the process of anaerobic glycolysis. In case of irreversible cell injury, these changes become more severe and there are some additional changes. Severe cell membrane damage causes the leakage of intercellular enzymes and protein into the circulation and entry of calcium into the cell that causes further damage of the cell. Marked mitochondrial dysfunction, and there are some large amorphous densities seen within the inner mitochondrial matrix and irreparable damage of the mitochondrial oxidative phosphorylation pathway that causes complete failure of production of ATP. And most importantly, within the lysosome, the lysosomal membrane damage causes leakage of lysosomal digestive enzyme into the cytosol that causes digestion of intercellular organelle, that means autolysis of the cell. And lastly, there are some nuclear changes. We call them pycnosis, karyoresis and karyolysis. We will describe them later. Now come to the details of irreversible cell injury. There are two types of irreversible cell injury. These are necrosis and apoptosis. Now we are discussing necrosis. During the process of injury, when a cell crosses the point of no return and progresses to cell death, there are some morphologic changes occurs during this process and after the death of the cell. These morphologic changes are collectively called necrosis. By definition, necrosis refers to a spectrum of morphologic change that follows the cell death in living tissue, largely resulting from progressive degradations of enzyme on the lethally injured cell. So what are the enzymes? These enzymes may come from cell's own lysosome or they may come from activated leukocytes as a part of inflammatory process. When the cell's own lysosomal enzyme causes the cell death, we call them autolysis, and if this process occurs by enzymes of activated leukocytes, we call them heterolysis. Now come to the morphology of necrosis. First of all, necrotic cell shows increased eosinophilia in the routine hematoxylin and eosin stain. This occurs due to the loss of cytoplasmic RNA. Secondly, the cytoplasm becomes homogeneous. As we know, the normal cell is heterogeneous, but due to the loss of glycogen particles, it's become more glassy and homogeneous in appearance. Thirdly, the cytoplasm become vacuolated and look like moth-eaten, reflecting the digestions of intercellular organelles and structural protein. Phospholipid of the cell membrane and organelle membrane are found accumulated, and we call them myelin figures. Under electron microscope, the necrotic cell is characterized by discontinuities of the plasma and organelle membrane, marked dilatations of the mitochondria with appearance of large amorphous densities, intercytoplasmic myelin figures, amorphous debris and aggregates of fluffy materials representing denatured protein. Most importantly, the nuclear changes occurs in the form of one of the three patterns. First of all, the basophily of the chromatin may fade. It is called karyolysis. Secondly, the nuclear shrinkage may occur with the increased basophilia, where the DNA condensed into solid shrunken basophilic mass. It is called pycnosis. And thirdly, the another change is called the karyoresis, where the pycnosis or pycnotic or partially pycnotic nuclei undergo fragmentation. Now come to the classification of necrosis. There are two basic types of necrosis. These are the coagulative necrosis and liquefactive necrosis. There are some other special types. Now come to the basic types. First of all, the coagulative necrosis. The coagulative necrosis results from denaturation of structural and enzymatic protein due to increasing intracellular acidosis of the coagulated cell. As the protein denaturation occurs here, that means the protein has lost its natural characters. There are no chance of further proteolysis, so the normal cellular shape, outline and architecture is preserved, but the nuclear changes occurs as usual of injurious stimuli. Examples of this kind of injury includes the ischemic necrosis of the heart, kidney, liver, adrenal gland and solid organs. On the other hand, in the liquefactive necrosis, the complete digestion of cell occurs and the necrotic tissue transformed into liquid viscous mass. It is characterized by bacterial or fungal infection because this agent stimulates the inflammatory cells. Examples include the abscess, boil and ischemic necrosis of the brain.